Welcome to Methods in Biostatistics. Today, we are going to explore one of the most fundamental concepts in all of statistics, the relationship between a population and a sample. Whether you are studying a specific species of fish in the ocean or analyzing clinical trials for a new medicine, understanding how to gather and interpret data is essential. Let us dive in and see how we can make sense of massive amounts of information using just a small, carefully chosen group. To begin, imagine you are cooking a massive pot of soup for a large family gathering. To check if the soup has enough salt, you certainly do not need to drink the entire pot. Instead, you stir it well and taste just one small spoonful. If that single spoonful is perfectly salty, you can safely assume the whole pot is perfectly salty. This simple, intuitive idea is the exact foundation of biostatistics. We study a small, manageable group to understand a massive, unmanageable group. Now that we have this intuition, let us define these terms formally. Building on our soup analogy, the entire pot of soup represents what we call the statistical population. In biostatistics, a statistical population is the complete, entire set of individuals, items, or data points that we want to study and draw conclusions about. It represents every single member of a specific group. For instance, this could be all the diabetic patients in a country, or all the oak trees in a specific forest. However, as you might guess, measuring every single member of such a massive group is usually impossible. This brings us to our next crucial concept. Because we can't possibly measure every single member of a population, we must select a sample. Think of the sample as that single spoonful of soup. A sample is a smaller, representative subset, carefully chosen from that larger population. The key word here is representative. Just as you must stir the soup before tasting it, a sample must accurately reflect the diversity of the entire population to be useful. By analyzing this smaller subset, we can draw highly accurate conclusions about the whole group without needing to examine every individual. Now that we understand populations and samples, we need to distinguish between the measurements we take from them. When we calculate a measurement from the entire population, we call it a parameter. You can remember this because both population and parameter start with the letter P. On the other hand, when we calculate that exact same measurement from our smaller sample, we call it a statistic. Both sample and statistic start with the letter S. This distinction is critical because our ultimate goal in biostatistics is to use a known sample statistic to estimate an unknown population parameter. You might wonder, why do we even need to separate populations and samples? Why not just measure everything? The primary reason is practicality and resource management in real-world scientific research. Measuring an entire population is almost always too expensive, too time-consuming, or physically impossible. For example, a marine biologist cannot possibly catch and weigh every single fish in the ocean. Furthermore, some testing is destructive. If you want to test the lifespan of a batch of medical syringes, testing the whole population would leave you with no syringes to use on patients. Sampling solves these problems elegantly. Now that we know why we sample, to keep things organized, statisticians use different mathematical symbols, depending on whether we are talking about a population or a sample. As a general rule, we use Greek letters for population parameters and standard English letters for sample statistics. This visual separation helps prevent confusion when reading complex research papers or performing calculations. When you see a Greek letter, you instantly know it refers to the true, exact value of the entire group. When you see an English letter, you know it is an estimate derived from a smaller subset. Let us look at how this applies to calculating an average. Let us look at the mathematical form of this concept. Here, we have the formula for the population mean. Conceptually, this is the true, exact average of the entire group. Let us break down the symbols. The Greek letter mu represents the population mean itself. The capital English letter N represents the total number of individuals in the entire population. The expression x sub I represents each individual value, and the capital Greek letter sigma tells us to add all those individual values together. If the population size n increases, it means we have more individuals to measure, making this true mean much harder, or even impossible to calculate in real life. Because the population mean is often impossible to find, we rely on the sample mean. The math is exactly the same, adding up the values and dividing by the count, but the symbols change to show we are looking at a small piece of the group. The symbol x bar represents the sample mean. The lowercase n represents the sample size, which is the number of individuals we actually measured. The lowercase x sub I represents each individual value within our sample. By comparing x bar to mu, we instantly know whether we are looking at our small sample or the entire population. Let us see this in action. Before we move on, let us see this formula in action. Let us imagine a small, isolated pond that contains exactly 50 frogs. A biologist wants to estimate their average weight, but only has time to catch a sample of 5 frogs. The weights of the caught frogs are 20, 22, 19, 24, and 20 grams. We need to calculate the sample mean. First, we add up the weights of the 5 frogs we caught to find the total mass, which gives us 105. Then, we divide that total sum by our sample size of 5. The resulting sample mean is 21. Physically, this means we estimate the average frog in the entire pond weighs about 21 grams. Now, suppose the biologist decides that a sample of 5 frogs is too small to be reliable. They go back to the pond and catch a larger sample, this time catching 10 frogs. The sum of the weights of these 10 frogs is found to be 215 grams. We will again use the sample mean formula, but this time our denominator has increased from 5 to 10. By dividing the new total weight of 215 by the new sample size of 10, we get a new sample mean of 21.5. Notice that this number is slightly different from our first estimate. Why did our sample mean change from 21 to 21.5? In biostatistics, the size of your sample directly impacts the reliability of your results. As your sample size, lowercase n, increases and gets closer to the total population size, capital N, your sample statistic becomes a much more accurate reflection of the true population parameter. The larger sample of 10 frogs gave us a better estimate of the true average weight of all 50 frogs in the pond than the smaller sample of 5 did. Let us visualize this relationship between the population and the sample. To tie all these concepts together, here we have a visual representation of the entire process we have been discussing. On the left, you see a large circle filled with many dots. This is our statistical population, with its total size, capital N, and its true parameter, mu. Because it is too large to measure completely, we follow the top arrow to extract a smaller subset, shown as the smaller circle on the right. This is our sample, with size lowercase n and statistic x bar. The most important part of this diagram is the bottom arrow, labeled inference. It shows how we take the math we did on the small sample and project it back to make an educated guess about the entire population. Let us review the key takeaways from today's topic. First, a statistical population is the entire group you want to draw conclusions about, while a sample is the specific, smaller group you actually collect data from. Second, we use Greek letters, like mu, for population parameters, and English letters, like X bar, for sample statistics. Third, we use samples because studying entire populations is usually impossible or highly impractical. Finally, always remember that calculating statistics from a larger, well-chosen sample will give you a much clearer and more accurate picture of the whole population. You now have a solid foundation in statistical sampling.